Hi, my name is Jessica Davenport and I'll be doing my presentation on the rabies virus. This first slide shows the scientific name of the rabies virus. So the order is Mononegavirales and the family is Rhabdoviridae with a genus of Lyssavirus, and the disease caused is rabies. This slide shows the characteristics of a rabies bacteria. I have pictured on the right a demonstration of those characteristics. So it has non-segmented negative stranded RNA genomes with a distinct bullet shape. The virus is approximately 180 nanometers long and 75 nanometers wide. There are 5 proteins encoded by the rabies genome, and that includes the nucleoprotein, phosphoprotein, matrix protein, glycoprotein, and polymerase. It also contains a helical ribonucleoprotein core and a surrounding envelope. So on the right, I have a cross-sectional view of that bacteria so that you can see those pieces demonstrated in the photo. The symptoms of rabies are very wide in range. So the earliest symptoms are typically very similar to those of the flu. So that could include fever, general weakness, headache, nausea, and vomiting. There also could be discomfort or itching at the site of the bite, assuming that that's how the disease was transmitted. There is a quick progression within days to symptoms of cerebral dysfunction, anxiety, confusion, and agitation. And the later symptoms of the disease may include delirium, abnormal behavior, hallucinations, and insomnia. The acute period of this disease ends after 2 to 10 days, and there also is a possibility of an incubation period after the bite. So that incubation period is the time between the bite and the appearance of symptoms. It could last for weeks or months. And what's interesting about this is that a bite from an animal during the incubation period doesn't carry a risk of rabies because the virus has not made it yet to the saliva. In animals, the diagnosis is done by a direct fluorescent antibody test. So that test looks for the presence of rabies virus antigens in brain tissue. In humans, it's a little more complicated and takes several tests to determine. There is a need for rapid and accurate diagnosis for the ability to treat this disease effectively. Lab tests for rabies are able to be standardized, fast, specific, cost-efficient, and reliable. Treatment begins with immediately and thoroughly cleansing the wounds, and this is for whether the person that was infected had previously been vaccinated or not. If that person was previously vaccinated, the next step should be to receive 2 IM doses at 1 milliliter each of the vaccine for rabies. 1 dose would come immediately after the infection was encountered, and the other dose would be 3 days later. For a person who was not previously vaccinated, they would have 4 1 milliliter doses of HDCV or PCEC available. This would be administered intramuscularly, and the first dose would be administered also as soon as possible after the exposure. The other doses would be administered on days 3, 7, and 14 after the first vaccine was administered. And the unvaccinated person should also receive rabies immune globulin once at the beginning of the anti-rabies prophosphorylis. And this would not be the same for a person who has been previously vaccinated. Once the symptoms of rabies appears, it is almost always fatal. So at that point, the treatment becomes supportive rather than trying to actually get rid of the disease. And there are less than 10 documented cases of human survival from clinical rabies. But the good news is that there is a preventative vaccine. So if everybody were to get that preventative vaccine, that could really help avoid needing a treatment at all. And that disease prevention includes the administration of both passive antibody through an injection of human immune globulin and a round of the injections with this rabies vaccine. So typically, humans contract rabies from the bite of a rabid animal, sometimes from infectious material of a rabid animal. So an example of the infectious material would be if saliva or brain tissue got directly in a human's eyes, nose, mouth, or open wound. There also is a possibility of the inhalation of aerosolized rabies virus, which is not commonly encountered. The only time that this would really be seen would be in a laboratory. Human-to-human transmission is very uncommon, but has occurred in 8 cornea transplants that have been documented and 3 solid organ transplants. The reservoirs could include all species of mammals because they are all susceptible to the rabies virus, but there are only a few that are really relevant to us humans. So in the US, the distinct strains have been found in raccoons, skunks, foxes, and coyotes, and also a few species of bats. And that is the end of my presentation.